Okay, so this is part two of my protein synthesis videos. And we're going to look at what we do with that messenger RNA that was made in transcription. Like, how do we translate it and use those directions that are in a nucleic acid in a series of nucleotides? How do we get that into an actual protein or polypeptide made out of amino acids? Okay, so now we're looking at this step. How do we build a protein? So in eukaryotes, we just saw how mature mRNA is made by the RNA processing, and that mature mRNA is going to leave through a nuclear pore to a ribosome, whether that ribosome is on the rough ER or in the cytoplasm. The ribosomes are going to read that messenger RNA to build the protein. Now, translation in prokaryotes is going to happen pretty much simultaneously with transcription, and we'll see why towards the end of this video. Okay, so the messenger RNA leaves and goes to the cytoplasm. So here we have our mature mRNA. We see that 5' cap with the modified guanine. We see our polyadenine tail. So what's going to happen first is basically a part of the ribosome, the small ribosomal subunit, is going to come and attach. And so what we are looking at is basically like how a ribosome is going to read the messenger RNA to build a protein. Like, that is our big picture. And when this ribosome reads the messenger RNA, it's going to read it in groups of 3, like a triplets. So you may like, it may be referenced as like, oh, the triplet code, and we call these these group of 3 codons. Oh my gosh, Mary. We call these group of 3 nucleotides codons. So a codon is a group of 3 bases, and a codon is going to code for an amino acid. So here, I did it in yellow. It's hard to see. But here you have our first codon. Our first start codon is AUG here. So AUG is our first codon, and that will actually code for the amino acid met or methionine. Then up in our mRNA strand, our next codon is ACA. So ACA, and that will code for threonine, I think is how you say it, the amino acid. So what we use to figure out like how to read the codons in groups of 3, this table here is called the genetic code. Now, this is the code for all life. It is universal. Every single cell on Earth that does protein synthesis uses the same genetic code. This is what makes genetic engineering possible. This is why you can take like a firefly gene that glows and like put it into like bacteria. And so because the genes can be expressed by different organisms. So anyway, let's go back to that first codon. This is called our start codon, and this is how you read the genetic code. So here our first codon is, our first base is A. So we're going to look, this side here is our first letter. So that means we're going to use that left side first, and we find A. That means the amino acid that this codon codes for is going to be found somewhere in this row. Then our second base in the codon is U. So now here we have where the red and the blue lines cross, that box is where we're going to find our amino acid. And then our third letter, our third base is G. So where all 3 intersect, there you have AUG, and that's coding for the amino acid methionine or MET. Okay, so let's go ahead and, so this is true for all codons on how you read them to find the amino acids. Let's for fun do that second one that was ACA. So I go, again, I'm using an A, and then I go C, and then A, so here I have that threonine. Now you also notice there's some stop codons. So stop codons will help to signal the end of a protein or a polypeptide. So let's go ahead and see how translation works. So our first step is initiation. So here I have my messenger RNA with my codons, my groups of 3 bases, but my start codon, it's always the first codon, is AUG. So our first thing that will happen is we have a small ribosomal subunit. The bottom part, the small part of the ribosome will attach first to the messenger RNA. And then we have a new kind of RNA called tRNA that stands for transfer RNA. And it's transferring, or it's bringing amino acids to the ribosome, because ribosomes build proteins, and proteins are made of amino acids. So we got to get the amino acids to the ribosome. But which amino acids? Those are the directions in the messenger RNA. So now here we have our tRNA, and we have our amino acid at the top of our tRNA. And so what's going to go down is you have your large ribosomal subunit will attach now. And now let's go ahead and look at the remaining codons. So this is where you would use that genetic code table to figure out which amino acid is next. You would use that table and go C-A-U, and you would find out which amino acid is attached to the top of that tRNA. And so then, if you learned about macromolecules already, we learned that when you build a protein, the first step is building that primary sequence or that primary structure that's based on the code in DNA. Well, here it is. We're building peptide bonds by dehydration synthesis within the ribosome. So now an empty tRNA will leave. The ribosome moves down to the next codon. You have the next arriving tRNA carrying an amino acid. Which amino acid it is, that red circle, you would read that table and go GAG and find the name of that amino acid. Then a peptide bond will form by dehydration synthesis between the two amino acids. Then the ribosome is going to move to the next codon. And as it moves to the next codon, then the next arriving tRNA can come in. A peptide bond will form, etc. So now our next codon here, ACG, we have a new tRNA carrying the next amino acid. A peptide bond will form, and that tRNA will leave. Now, I should mention the tRNAs that leave, like this one right here, it's empty. Well, it's got to get a replacement amino acid, right? And this is why we eat. Half the amino acids, about 10 or so of our amino acids, we can make ourselves, synthesize ourselves. And the other half of our amino acids we have to get from our food. This is why it's important to eat a wide variety of meals and items in your food. In places in the world where there's high amounts of poverty and people eat like just one food, like maybe rice or a particular grain, and it's missing amino acids, they become like malnourished. And so that would be because they're missing particular amino acids in their diet. Okay, anyway, so now our very end of our mRNA is what we call a stop codon. So here, instead of a tRNA attaching, we have what's called like a releasing factor will come and basically it signals the end. So at this point, when a stop codon is reached, then everything breaks apart and you have your cute little polypeptide. Now right here, remember a polypeptide is a chain of amino acids. This is only 5, but in reality, on average, like a human protein is about 400 amino acids. So to summarize or to point some key things out, here we have our messenger RNA. In our messenger RNA, we had 5 codons. We always start with AUG. Now, sometimes there might be amino acid or nitrogen bases upstream, before AUG that are just non-coding, or what's it called, untranslated, like they're not translated, they're not turned into amino acids. So you want to find your start codon. Wherever AUG is, that's where you start. And from there, you can begin translation. And so eventually, and there's a stop codon. Now here we have 5 codons plus that stop codon and 5 amino acids. So there's 15 bases that coded for those 5 amino acids. And then we had our 3 for our stop codon. So sometimes I've seen some like critical thinking questions that will ask like, how many amino acids is in a polypeptide? Oh, my question's not even good. I meant to say how many nitrogen bases, how many nucleotides is in a polypeptide made from 219 amino acids? My question's bad. Just ignore that. Sorry. I think I was watching a Christmas movie with my kids when I typed that. Anyway, so let's go ahead and if you're my student, I'll give you better questions in class. Sorry. All right, so now let's go ahead and look at our transfer or our tRNA in a little bit more detail. So tRNA is RNA, and we learned that RNA is single-stranded. So when we look at our tRNA, it is actually a single strand of RNA that kind of folds like onto itself. So like right here, these are actually like base pairs, like hydrogen bonds between itself, which is kind of neat. And it does form this like four-leaf clover or this like T shape. So an important area though on this tRNA is the bottom region. This bottom region is called an anticodon. And the anticodon is kind of cool because now let's look at like this mRNA. The mRNA, here we have our codon in our messenger RNA, and that actually pairs with the anticodons on the tRNA. It's like how the tRNA, when it arrives in the ribosome, there's some hydrogen bonds that form between the anticodon and the codon, and that's how it kind of gets like stabilized long enough for the peptide bonds to form between the amino acids. Now, it's at the top of the tRNA is where you find the amino acid. Okay, so our last little conversation here, I think, is about translation in prokaryotes. I want to point out eukaryotes, their two steps, transcription and translation, are separated like physically by a nuclear envelope, right? They have the nucleus and the cytoplasm, but prokaryotes don't have that. Also, in eukaryotes, we have RNA processing. That doesn't happen in prokaryotes. So here, in prokaryotes, when RNA polymerase attaches, you also notice there's no transcription factors here. Okay, so when RNA polymerase attaches and it transcribes, basically a ribosome can begin translating right away. So those little yellow circles represent the amino acids. So here you have transcription followed right away by translation. So they kind of happen simultaneously. And you can have more than one ribosome attached onto this messenger RNA. They can have multiple ribosomes attached to the mRNA. All right, so let's summarize our types of mRNA and what we are just RNA in general. So here's our nucleus. We have our chromatin and our nucleolus. The nucleolus, that really dense region, is where our RNA is made, and we haven't talked about that yet. And then we have our nuclear pore, and you can see how that that's where the RNA leaves the nucleus. It goes through the nuclear pore. Okay, so inside of our nucleus, we have a really dense region called the nucleolus. It's still part of our DNA. It's our nucleus. It has our chromatin there. So the nucleolus, though, is the region where there's transcription happening to make a type of RNA. We haven't learned that yet, but rRNA actually stands for ribosomal RNA, and it's used to build ribosomes. So ribosomes are made out of rRNA and proteins. And so we have rRNA that makes ribosomes. Then we have our messenger RNA. So our messenger RNA is made to make a copy of our directions on how to build a protein to carry it from the nucleus to a ribosome out in the cytoplasm. And then we have our tRNA. Our third type of RNA is called transfer RNA, and that's basically what brings our amino acids to the ribosome. All right, so this concludes my video on translation, because I think I'm going to do a separate video. So our three types of RNA are here, and they're all made by transcription. My next video will talk about the endomembrane system and the organelles involved in this, as well as the different kinds of ribosomes. All right, good job.